Hello students, welcome to this chapter. In this chapter, we are going to study genetics. It can be classical genetics and molecular genetics. Genetics is a branch of biology that studies about heredity. Heredity means the pass of genetic information from one generation to the next generation, sexually or asexually. In this lesson, we are going to study genetic cross. Before we are going to study genetic cross, let us see some vocabulary words. These vocabulary words include the first one is gene. Gene is a section of DNA and therefore a section of chromosome that determines a particular feature. The second vocabulary word is allele. Allele is an alternative parts or versions or different forms of a gene that determines a particular contrasting features. The third one, what is chromosome? Chromosome is a long strand of DNA on which tremendous number of genes are stored and is wrapped itself around the histone protein. Homologous pair. Homologous pair is pairs of similar chromosomes which have the same gene sequence for the same features at the same position or location. Locus, plural, loci, is the position of a particular gene on a chromosome. Homozygous means the situation when the same alleles of a gene present in homologous pair per locus. Heterozygous is the situation when different alleles of a gene present in homologous pair per locus. What about genotype? Genotype describes pairs of alleles for a particular feature possessed by an organism or the internal genetic setups of an organism. Phenotype describes traits or external observable features or physiology that is determined by genotype. Monohybrid cross or monohybrid inheritance. This is a genetic cross or breeding experiment that relates to just the inheritance of one trait or feature. The father of genetics, the man who discovered the rules by which genes are inherited, was Austrian monk Gregor Mendel. This person lived and experimented in a monastery called Brno, that's found in Austria. Mendel was highly methodical or meticulous experimenter. Mendel experimented with pea plants and able to deduce the rules of inheritance from his results. He noticed that pea plants exhibited contrasting characteristics. The 7 contrasting characteristics of pea plants. Mendel used the 7 contrasting characteristics of pea plants for his experiment to deduce principle of inheritance. These 7 contrasting features of pea plants are height, that can be tall and dwarf, flower color, which can be purple and white, flower position, which can be axial and terminal, seed color, yellow versus green, seed shape, round and wrinkled, pod color, green and yellow, pod shape, smooth and constricted. From these 7 contrasting features, the first variants of features are dominant traits or dominant features. Tall, purple, and axial are dominant features, whereas dwarf, white, and terminal are recessive traits. Here, yellow, round, green, and smooth are dominant variants or dominant traits, whereas green, wrinkled, yellow, and constricted here are recessive traits. Mendel did experiments on pea plants by crossbreed two pea plants with contrasting features. Pea plants has contrasting features or non-continuous variables. This was the key that enabled Mendel to experiment successfully. That means there were never medium height pea plants or plants with pale purple flowers. So, how did he do the experiment? Before he carried out any breeding experiments, Mendel self pollinated pea plants for several generations with regard to seven contrasting features to make sure that the pea plants are true breeding or pure line for that characteristics. Mendel was very methodical. His starting point was to wonder what would happen if he crossbreed two pea plants with contrasting features. For example, plants with purple flowers and plants with white flowers. Before he carried out any breeding experiments, he self pollinated the plants for several generations to make sure that the plants are pure line or true breeding for that characteristics. And eventually, he used plants from breeding line that had contained only purple flowered plants or white flowered plants. This he called true breeding plants. Self pollination means transferring pollen from anthers to the stigma in one parent. So he transferred pollen grain from the anthers of that parent to stigma of that parent. He then crossbreed them in the following way. We will use flower color as our example, but Mendel used the same procedure for all the seven contrasting features of pea plants. In our example, Mendel removed stamens from the flowers of the purple flowered pea plants so that these flowers could not pollinate themselves. That means there is no self pollination here. Second, he used paintbrush to transfer pollen from the flowers of white flowered plants to the carpel of the purple flowers. This pollinated carpel then produced a pea pod containing several pea seeds. He collected and grew all the seeds from all the pods. When the plants were matured, he noted the color of their flowers. In his result, all the offspring, which we call the F1 or first filial generation, have purple flowers. Mendel also carried out reciprocal crosses. In this case, he also pollinated white flowered plants with pollen from purple flowered plants. You can follow the reciprocal procedures what we saw in the previous procedure. In the above cross, in either procedures, all the offspring in the F1 or first filial generation have purple flowers. Mendel then allowed these purple flowered plants to self pollinate themselves. In the next generation, or F2 or second filial generation, he found a ratio of very nearly three purple flowered plants for every one white flowered plant. This pattern repeated itself in all of his experiments. In each case, he crossed pure breeding plants with contrasting characteristics. He found that only one of the characteristics appeared in the F1 generation, always, for example, purple flower, never white flowered pea plants. He found a ratio of 3 to 1 in the F2 generation, always 3 of the one that had appeared in the F1 and 1 of the one that had never appeared in the F1 generation. That means 3 purple flowered, for example, ratio 1 white flowered pea plants. It was his pattern that led Mendel to formulate his laws and to coin the terms dominant and recessive. He used the term dominant to describe the allele that determined the trait that appeared in the F1 and the term recessive to describe the allele that determined the trait that did not appear in the F1 generation. We can predict phenotypic ratio of Mendel's experiment by using Punnett square. Mendel crossed purple flowered pea plants capital P with white flowered pea plants small p. He then obtained all or 100% purple flowered pea plants capital P small p. When capital P crossed with small p, the gamete capital P, the gamete for the white flowered pea plants small p, capital P small p, capital P small p, capital P small p, capital P small p. All are capital P small p. Genotypic ratio is capital P small p. Phenotypic ratio is all or 100% purple flowered. Mendel then self-pollinated these purple flowered pea plants or F1 generation. In this case, in F2 generation, he obtained 3 purple flowered pea plants and 1 white flowered pea plants. When F1 all purple flowered pea plants self-pollinated, that means capital P small p crossed with capital P small p. When their gametes fused randomly, they could produce one capital P or purple, or two capital P or purple, or one small p white flowered pea plants. That means 3 or 75% purple and 1 or 25% white flowered pea plants can be produced in F2 generation. Gregor Mendel, after he did his experimental works on the 7 contrasting features of pea plants, he combined his results in table 3.1. In this experimental procedure, we can see the 7 contrasting characters: flower color, flower position, seed color, seed shape, pod color, and stem length. These 7 characters have its own contrasting features: the dominant trait and recessive trait. The dominant traits are purple for flower color, axial for flower position, yellow for seed color, round for seed shape, inflated for pod shape, green for pod color, tall for stem length, and recessive trait includes white, terminal, green, wrinkled, constricted, yellow, and dwarf. After he cross-pollinated them, he obtained the ratio of number of individuals of dominant versus recessive. For flower color, he obtained 705 purple flowered pea plants, ratio 224 white flowered pea plants, and for flower position, he found 651 axial plants, 207 terminal pea plants. For seed color, he obtained 6022, ratio 2001. For seed shape, he obtained 5474 round seeded pea plants to 1850 wrinkled seeded pea plants. In case of pod shape, he found 882 inflated pod-shaped pea plants to 299 constricted pod-shaped pea plants. In case of pod color, he found 422 green colored pod-shaped pea plants to 152 yellow pod-colored pea plants. In case of stem length, he obtained 787 tall pea plants to 277 dwarf or short pea plants. When we change this to ratio, he obtained
3.1521 in case of flower color, 3.1421 in case of flower position, 3.0121 in case of seed color, 2.9621 in case of seed shape, 2.9521 in case of pod shape, 2.8221 in case of pod color, and 2.8421 in case of stem length.
Then Mendel explained his results in the following way and developed certain principles of inheritance. The first one is trait controlling factor principle. In this principle, Mendel put forward an idea that every trait is controlled by two heritable factors or genes. The heritable factors or genes may take different forms, what we call alleles. If the two alleles in an individual are different, one is dominant and the other is recessive, what we call complete dominance principle. Mendel also explained that the only physical link between the generations is the gametes or sex cells. This must pass the genes from one generation to the next generation. The heritable factors or alleles separate when the gametes or sex cells are formed. Each gamete contains only alleles controlling the variants of traits. This is Mendel's law of segregation. He also stated that the gametes or sex cells fuse randomly at fertilization. Mendel also formulated another law, what he called the law of independent assertment. In the law of independent assertment, he stated that the inheritance of one trait is independent of the inheritance of another trait. The alleles of one pair segregates independently of the alleles of another pair controlling different feature when the alleles are non-linked. How could we find out if an organism showing the trait determined by a dominant allele is homozygous or heterozygous? The only possible way is to carry out a breeding experiment. This breeding experiment to find out if an organism is homozygous or heterozygous for a dominant trait is called test cross or back cross. Back cross or test cross is breeding experiment to find out if an organism is homozygous or heterozygous for the dominant trait. The dominant trait in monohybrid cross can have two genotypes, homozygous or heterozygous alleles. So, an organism showing the dominant trait must be bred with those whose genotype is definitely known. Organisms that have definitely known genotypes are organisms that have recessive traits because recessive traits always expressed when two recessive alleles of a gene appear per locus on homologous pair. Let us use flower color in pea plants as our example. As we can see from the Punnett square, we crossed purple flowered pea plants with white flowered pea plants. Genotypes of purple flowered pea plants could be capital P or capital P small p. So, how can we find out the genotypes of this dominant trait, purple flower? We crossed purple flowered pea plants with white flowered pea plants. If the genotype of the dominant trait is capital P, all of the offspring can be purple flowered. That means 100% purple flowered pea plants. If we acquired 100% purple flowered pea plants, we can be quite sure that the unknown genotypes for the dominant trait can be capital P, or homozygous. If the genotype of the dominant trait is capital P, small p, half of the offspring can be purple flowered and half of the offspring can be white flowered pea plants. That means 50%, 50%. If the phenotypic ratio is 50% purple flowered and 50% white flowered, the unknown genotypes for the dominant trait could be capital P, small p. If any of the offspring have white flowers, we can be certain that the original purple parent was heterozygous. If all the offspring are purple flowered, we can be almost certain that the purple flowered plant was homozygous. White flowered offspring, homozygous recessive, would need to inherit two recessive white alleles from each parent. But because predicted ratios are not always met in the real world, we couldn't be absolutely certain.